Every year, healthcare systems worldwide administer roughly 15 billion doses of injectable drugs. To ensure patient safety, regulatory bodies mandate that these sterile injectable products must be manufactured to be essentially free of visible particulates. A major friction point exists across the pharmaceutical industry regarding how field inspectors for agencies like the FDA actually interpret and enforce this subjective phrase. As a result of these strict field interpretations, nearly a quarter of all injectable product recalls are triggered by the discovery of particulate defects. These recalls are driven by a pervasive industry myth, the assumption that the compendial standard of essentially free is identical to a mandate for absolute zero particles. Treating essentially free as a requirement for absolute perfection creates a statistical impossibility. When manufacturers and regulators demand a zero-defect reality, they trigger massive lot recalls over single isolated particles, directly driving medically unjustified drug shortages for critical treatments. Evaluating these vials relies on visual inspection, which is not an absolute barrier. It is a probabilistic human function. A human inspector's ability to spot a defect depends on specific physical variables, including the particle's size, its specific shape and color, and its density against the fluid. Establishing a baseline for this inspection requires highly controlled, idealized conditions. Inspectors must view the vials under diffuse illumination, set precisely between 2,000 and 3,000 lux. Lux is a standard unit of illuminance that measures the amount of light falling on a given surface area. Because human sight operates within strict biological thresholds, even a process of 100% manual inspection of every single vial can never guarantee the total removal of all particulate matter. This chart shows the probability of detection, or POD curve. It graphs the human detection rate on the y-axis against the size of spherical particles, measured in micrometers, on the x-axis. If we trace the curve for a spherical particle measuring 50 micrometers across, the probability of a human inspector detecting it hovers near 0%. Moving up the curve to 100 micrometers, the detection rate improves, but it remains effectively a coin flip, sitting between 40 and 60%. The critical inflection point arrives at 150 micrometers. Only at this size does reliable visual detection of 70% finally begin under idealized conditions. To reach a 95% detection guarantee, the particle has to extend to 200 micrometers in diameter. This curve flattens out for non-spherical particles, like fibers. For these shapes, reliable visual detection does not even begin until the particle reaches 500 micrometers. Because humans cannot reliably see anything smaller than 150 micrometers, any qualified visual inspection system carries an inherent mathematical certainty. The vast majority of particles escaping detection and entering the supply chain will be subvisible. To manage this biological limitation, the United States Pharmacopoeia developed the USP 1790 lifecycle framework. This serves as the engineering solution to the reality of the probability of detection curve. Under this framework, the mandatory 100% visual inspection of a batch is treated merely as a primary high-volume filter. It is not designed to be the final guarantee of product purity. Instead, the system relies on acceptable quality limit, or AQL, sampling as the secondary safety net. AQL is the highest number of dissective units permitted in a sample size to still accept the overall manufacturing lot. This matrix charts AQL thresholds based strictly on defect severity. Critical defects receive the lowest allowable percentages. The matrix then flips to reveal the unacceptable quality limit, or UQL, which defines the true statistical risk of passing a defective batch to the patient. If a sample exceeds the AQL, it does not mandate an immediate recall. Instead, it triggers a root cause investigation or a complete reinspection of the lot. The entire system is explicitly designed to manage mathematical probabilities, rather than initiating blind recalls over isolated anomalies. Despite rigorous statistical controls, we must account for the clinical reality of a single subvisible particle surviving the filter and entering the human body. For medications administered through subcutaneous or intramuscular injections, the introduction of a sterile, inert particle typically causes minimal local tissue irritation, or at worst, a small, benign granuloma. Clinically significant risk only truly materializes when a particle bypasses local tissue and is introduced directly into the bloodstream through the intravenous route. To understand that risk, we have to look at vascular mechanics. The smallest blood vessels in the adult human body are capillaries, which measure roughly 7 micrometers in diameter. This chart shows how that baseline compares to the respiratory system. A pulmonary capillary ranges slightly larger, from 10 to 15 micrometers across, making it entirely dwarfed by a 150 micrometer visible particle. This creates a strict mechanical occlusion threshold. Any rigid, undissolved intravenous particle larger than 10 micrometers will eventually physically lodge tightly into a capillary bottleneck. Conversely, particles smaller than 7 micrometers will generally pass through these bottlenecks, where they are phagocytized by macrophages. Macrophages are specialized white blood cells that engulf and digest cellular debris, ultimately depositing these microscopic particles safely in the liver or spleen. But an occlusion from a larger particle does not automatically equal patient harm. When a single particle blocks a tiny vessel, these physical capillary occlusions rarely manifest as detectable symptoms in the patient. The reason is vascular reserve capacity. Human organ systems possess massive anatomical redundancy, meaning multiple branching blood vessels constantly supply the exact same tissue. We can see this tolerance clearly in the historical precedent of extracorporeal circulation during open-heart bypass surgery. In these procedures, patients routinely tolerate a significant volume of solid particle microemboli generated by the bypass machinery without suffering long-term clinical consequences. Because of this extensive vascular redundancy, the human body easily reroutes blood flow. A single, isolated capillary occlusion from a microscopic defect remains entirely clinically silent. The negligible impact of an isolated defect stands in stark contrast to the severe physiological trauma caused by massive, chronic particle loads. Medical literature documents these massive loads in the extreme cases of illicit intravenous drug abuse. These patients frequently inject pulverized solid tablets mixed with unregulated, completely insoluble excipients. These chronic, high-mass exposures lead directly to pulmonary foreign body emboli and widespread systemic microvascular degradation. Similar damage appears in specific clinical extremes, such as total parenteral nutrition precipitation. When these high-volume nutritional fluid mixtures become unstable, they rapidly form thousands of precipitating crystal formations, delivering massive calcium phosphate infusions, triggering acute respiratory distress. Additional risk is found in severe anatomical edge cases, such as injections into the closed space of the intrathecal route around the spinal cord, or in highly vulnerable demographics, like premature infants receiving continuous fluids through umbilical catheters. Actual clinical danger requires a massive aggregate mass of particles or highly vulnerable patient physiology. These conditions are entirely divorced from the reality of a single rogue particle slipping through a sterile manufacturing line. When an isolated particle does appear, the physical threat of vascular occlusion is secondary. The true underlying threat of particulate matter is the potential risk it poses to the sterility assurance of the drug product. To assess this, investigators classify the material. Intrinsic particles are materials originating from the known manufacturing process itself, such as glass lamellae peeling from the vial or rubber shedding from the stopper. Conversely, extrinsic particles are exogenous materials completely foreign to the process, such as insect parts, human hair, or non-processed cellulose fibers. Extrinsic particles carry an infinitely higher risk profile. Because their origin is unknown, they possess an uncontrolled bioburden and a high potential to leach toxic chemical compounds directly into the liquid medication. This makes forensic identification of any discovered particle mandatory. The biological threat of microbiological contamination far outweighs the physical threat of a microscopic capillary occlusion. Decades of medical literature show virtually zero documented patient harm resulting from the injection of a single, sterile, inert visible particle. When a single particle is discovered in the field, the response protocol must trigger a contextual risk assessment rather than an automatic mandatory lot recall. This assessment requires investigators to evaluate the manufacturing facility's historical trending data, conduct strict forensic particle identification to rule out sterility risks, and account for the target patient demographics of the specific drug. The compendial requirement to be essentially free of particles is a standard based on manufacturing probability and statistical safety. Aligning regulatory enforcement with actual medical risk ensures that a pursuit of absolute zero does not sever global patient access to critical, life-saving treatments.